Hi. First, thank you for coming here and listening to our presentation. So in this presentation, we will introduce about biotoxicity. So biotoxicity means how substance being harmful to living organisms. I mean, things like bacteria that giving disease to human being a problem for a long time. So scientists studied about how we can suppress this bacterial growth. And I mean, some people will ask, why do we have to study all this? Because we have many medicines right now. But when we related this to nanoscience, it became a different story. Because nanoparticles are so small that they barely make a bad impact to patients. So this allows doctors to cure their patients without any side effects. So scientists found the one special substance that can kill cells really effectively, which is silver. And in biotoxicity lab, we measured how different forms of silver particles kill cells effectively. So the way we did is we prepare the yeast cells and then mix those with different form of silvers, which are ions, powders, and nanoparticles. So cells also breathe like a human, and they produce CO2, which is carbon dioxide gas, when they breathe out. So if particles can kill those cells well, they will not able to breathe well, which means they will produce less gas. But if particles are not toxic, they will not able to kill cells well, and cells will produce a lot of CO2 gases. So this picture summarizes the biotoxicity experiment. On the left side, 3 people are shooting arrows, and each person shooting different kinds of arrows. So the tips of arrows have a different size. The bottom one has the smallest, top one has a medium size, and middle one has a biggest size. And on the right side, someone drew the HP bar. They described how much damage they got. Well, it's I don't like that. It's so violent.
Okay, so
Hold on. However, so this picture will describe how different forms of silver will give different amount of damage to yeast cells. So
we did an experiment on how different types of silver particles will damage cells. So in our experiment, we as he said, we used silver powder, silver nanoparticles, and silver nitrate. So silver powder had the largest particles, but since those particles couldn't enter the cells, they weren't able to deal damage. The silver nanoparticles had medium-sized particles, and they could enter the cell and deal a good amount of damage. Lastly, the silver nitrate had the smallest particles, but even though they could enter the cell and deal damage, they couldn't enter the cell very quickly, so they weren't able to deal that much damage. This picture is analogous to the different forms of silver particles because on the top the arrow had a medium tip and it was able to deal the most damage. On the second image, the arrow had a really large tip, but it wasn't able to really deal damage, kind of like how the silver powder wasn't able to damage the cell. And for the third arrow, it had a really small tip, kind of like the silver nitrate, and it was able to deal some damage, but the person still lived.
Okay. First of all, I will start by correcting some of the things that my partner said. Long before there were any scientists, ancient Romans discovered that the silver had some antimicrobial. Yes, ancient Romans used jugs made of silver to store water and milk to prevent bacterial growth, and in later times, even in the U.S. People started putting U.S. silver coins in milk to prevent bacterial contamination. And my partner said, size does matter. In silver particles, they may take on a silver may take on different sizes, and say in both sizes, their antimicrobial effect might not been so effective, and but as nanoparticles, their sizes are small enough to enter cells and enough to disrupt cell functions and to break down the cell wall and thus killing the bacteria. And I would say the takeaway from this lab is that as we examine the size of the silver particles, it's important to know that small sizes or sizes at a certain scale does matter. So.
Okay, so one limitation of the piece is that it doesn't portray how different sizes of silver particles enter the cell differently. In the experiment, while the small silver nitrate entered the cells through protein channels, the medium-sized particles entered the cells through endocytosis. But in this picture, all of the arrows hit the person in the same way, which basically just doesn't show like the different methods of entrance.
All right, thank you.
All right, so I'll ask the first question. So I do agree that it does look like the method of entry is the same, but I do think that there is one specific thing about the top panel with the nanoparticle that is showing something that the nanoparticle does in the actual experiment. So can you see kind of, here I'll borrow the laser pointer for a second. So I think there's something that's happening here that is kind of a clue as to how this is also really analogous to a nanoparticle. Can you think of what that is?
I think it describes the surface area, because basically the basic concept of toxicity is the more surface area a particle has, they have more contact so that they can kill like more cells more effectively. So since nanoparticles are really small, compared to same volume, they have much greater surface area. So I think Painter explained it by a number of arrows.
Right. So when the nanoparticle enters inside the cell, then what does it do?
I'm pretty sure it harms the mitochondria of the cell, which limits the cell's ability to do cellular respiration. In our experiment, we saw that as the nanoparticles killed the cells, the cells started producing less carbon dioxide because they were dead, so they couldn't do cellular respiration anymore.
So what is the nanoparticle releasing when it's inside the cell that damages the mitochondria?
Well, in this case, our silver ion may lead to overproductions of certain oxygen, what is it called? Reactive oxygen species, which is also a process called liquid peroxidation, and that will lead to the basically the breakdown of the cell wall.
Right. Right. So I think that's exactly what's happening between this one and this one, right? So this one is, I think, the ion. That's a small arrow. And I think this is meant to be one arrow that, because it's lit on fire, gets split off into a bunch of small arrows, just like the small arrow of the ion down here. So I think that is one additional thing that the drawing was trying to convey, although the artist will tell us if that was their intent afterwards. Is there one other question?
Why did the artist make it violent?
Well, I guess that's partly for the artist to interpret, but I do think it's an analogy if you guys want to explain why they made it violent.
Personally, I believe this element of violence does relate to our common understanding, because it's just simple killing and slaughter. That's something we can relate to instead of just scientific jargon.
Well, I think what I might add to what you say is that they made it violent because the silver in all of these forms are damaging the cells. They're hurting the cells, right? They're killing the cells. So that is why they chose to use a similar type of analogy here.
Yeah,
So,
I would agree.
yes. All right, so let's thank our presenters. Good job.